Thank you very much for the kind introduction. My lab uses integrative structural biology to study biological CO2 fixation. And as you can see here on this rather beautiful artwork, we're dealing with some very exotic, one could even say alien microbes. Their metabolism is very different to what we understand from normal metabolism. And these are acidogenic bacteria. And even among bacteria, these are very special bacteria. They grow on a variety of different substrates. They grow on sugars, they grow on formate, methanol, lactate, in particular, they grow on gases alone. They grow on CO2 and hydrogen, and in all these cases, they produce acetate. That's the molecule that's responsible for their namesake. They are called acetogens. And acetate formation plays a key role in the anaerobic food chain. But not only. With a small biotechnological modification, one can convert acetogens to not produce acetate, but instead produce bioethanol. And they can be used as green fuels. And moreover, these green fuels are the center of a whole industry. So the next slide gives you an impression that we're not talking about small lab-scale reactors. We're talking about companies. We're talking about $billion in value. We're talking about big fermentation tanks in the thousands of liters. But enough of that for now. So where my lab comes now into play is that we want to understand the chemical basis, the enzymatic reactions. We want to understand the difficult chemistry. We want to understand this exotic mechanism, to not only have a solid understanding of biochemistry, but also to pave the way for biotechnological and synthetic biological applications. But back to my fascination. These acetogens, they can grow on gases. They produce acetate and another class of anaerobic microbes. They're called methanogens. They use CO2 and hydrogen to produce methane. And these classes of organisms together, they live under very extreme conditions. They live on high salt, down at the ocean. There is basically no oxygen. There's little nutrients, basically nothing to feed off, apart from hydrogen and carbon monoxide, which they can oxidize to get some electrons, and CO2, which they use for carbon fixation. And despite being very different, well, acetogens and methanogens, they belong to different kingdoms of life. They're bacteria and archaea. They share variants of the same basic metabolic pathway, the reductive acetyl-CoA or Wood-Ljungdahl pathway. But to just give you an idea how difficult it is to make a living on gases alone, we have a look at the thermodynamics of the equations. So under standard conditions, you can yield from 4 hydrogens and 2 CO2 roughly 95 kilojoule energy. This is not a lot, particularly if you compare it with 38 mole of ATP, which we get in aerobic respiration. And this basically is even more extreme if we look at the physiological conditions where these bugs live. So hydrogen is a rare commodity where they live. Every organism competes for hydrogen, and therefore the energy which is released, it's not even 1 mole of ATP. It's rather only a third mole of an ATP. And this is as low as you can get with metabolism. So these bugs, they live literally at the thermodynamic limit of life. So what is this metabolism which allows this? This is the Wood-Ljungdahl pathway. And in theory, it's the simplest way to fix a molecule of CO2 to carbon. And this is basically by simply putting 2 molecules of CO2 together to acetyl-CoA, similar to duplo bricks. It's a linear pathway that has 2 branches. First, the metal branch, where a molecule of CO2 is fixed to formate and then stepwise reduced to a metal group. And this activated metal group is then condensed together with a molecule of carbon monoxide to acetyl-CoA. And this is, as you all know, the turntable of the metabolism. It can be used for biomass assimilation. So you can make sugars, you can make out amino acids, you can grow on it. Let's have a look what happens in acetogens. Because acetogens, as the name says, they produce acetate. So in the first step, a molecule of CO2 is reduced by an enzyme called hydrogen-dependent CO2 reductase to formate. This formate then is an ATP-dependent step transferred to a C1 carrier called tetrahydrofolate. And then it's stepwise reduced by 2 molecules of NADH. And this metal group is then condensed with the carbon monoxide molecule that is formed here in a carbon monoxide dehydrogenase complex by the reduction of ferredoxin. And it's an enzyme-bound intermediate. So the carbon monoxide doesn't leave the enzyme, but instead stays there in one domain and attaches to the metal group, and is then condensed by a very special cofactor containing 2 nickel atoms to acetyl-CoA. And acetogens now use acetyl-CoA, transesterize it with a phosphate. The phosphate anhydride is energy rich, ATP can be produced, and acetate secreted. But we gain 1 ATP, but we need to spend 1 ATP. So the entire pathway is there to produce acetate, which they secrete, so they don't want it. So what is it all about? The answer lies in the 2 molecules of NADH here. So in acetogens, CO2 fixation basically serves as the terminal electron sink. So that means you get rid of reduction equivalents. One could even say they replace oxygen in an anaerobic metabolism with CO2 fixation. So let's have a look at the total picture, at the respiration. In acetogens, respiration is conceptually simple. It consists of only 3 proteins. The first one is the electron bifurcating hydrogenase. It oxidizes molecular hydrogen to produce NADH and ferredoxin at the same time. Some of the ferredoxin is now used to drive a membrane-bound respiratory enzyme, termed the RNF complex. So the RNF then produces NADH. So here we have our 2 molecules of NADH. They are used here in the CO2 fixation reaction. The sodium pumped by the RNF complex is then used to fuel the sodium-dependent ATP synthase. That's it. It's a minimal respiratory chain. And for this to work very well, it needs to run very efficiently and very specific. So these acetogens, they need to fix an awful lot of biological CO2. And they need to do it very well. And how do they do this? These are the questions we want to answer in our lab. This direct coupling between CO2 fixation and energy conservation is also one of the reasons why people think this metabolic pathway is one of the earliest that has been involved. So it's considered to be ancient. So what we're here looking at are the remnants of the evolution of life itself. So, but how do we study these enzymes? It's actually not that straightforward, because these are metalloproteins, and they like to bind gases, and they like to particularly bind oxygen, but they don't know oxygen. They haven't evolved in the absence of oxygen. So if they react with oxygen, they cannot cope with it. They denature. The cofactors disassemble, the organic metallic intermediates dissociate, and the proteins often they denature. So every working step needs to be taken care under fully anaerobic conditions. That's the first challenge. And the second challenge is we need to be able to get our proteins. And this is also not straightforward. You cannot use your recombinant E. coli. You cannot use insect cells. You cannot use everything which is standard production. You need to work with the organisms themselves. Either you need to do laborious multi-step native purifications, or you need to invest in genetics. And they are also not like E. coli. It can take up to half a year to clone one gene. But I have brave students, and they manage. And after all these challenging working steps, you still need to be able to do structural biology under fully anoxic conditions. So we developed a workflow which can do this quite efficiently. And as you can see here in the last basically 2-3 years, we managed to determine the majority of the respiration complexes in acetogens. Most of the work is published, a few of the works are in preparation or submitted. But I'm going to talk about 2 examples today. The first one is the hydrogen-dependent CO2 reductase. And I think this is an absolutely amazing enzyme. It's one of the most efficient CO2 fixation catalysts now today. It's 10,000 times more efficient than any known chemical catalyst and fully reversible at room temperature. And this attracts also biotechnological interest. So this is a cooperation together with Volker Müller in Frankfurt. He's an expert in acetogen genetics, and Ben Engel for cryo-electron tomography, as you will see in a minute. So we purified the HTCR complex from native source, so meaning without affinity purification. And then we could basically freeze it on grids, and what we saw here was a little bit of a nightmare. But after a lot of classification steps, we actually thought, okay, this enzyme forms short filaments. They are highly irregular, so bent, different length, different composition. But after a long struggle, we managed to get a 3-dimensional reconstruction of the complex. And the structure basically tells you already part of the answer. So here in blue, we have hydrogenases. In green, we have formaldehyde-hydrogenases. And here in the center, we have a filament of so-called polyferrodoxin-like proteins. And you can now see this actually has a lot of iron-sulfur clusters. And the iron-sulfur clusters, they connect the hydrogenase and the formaldehyde-hydrogenase with a direct electron chain. And they connect within the repeating molecular subunits. So what does this mean now? So we took our structure and had a look. What makes the filament formation? And we identified the C-terminal helix of one of these polyferrodoxin domains. So if you look at this protein, this is basically one bacterioferrodoxin fused to another bacterioferrodoxin, rotated by 180 degrees and put within each other in a sequence. So, and all these proteins have a C-terminal helix. And if we delete one of them, we can basically say, okay, we lose activity. And there are 2 isoforms, HIGB3 and HIGB4. And if we delete HIGB4, we dissociate within the repeating subunits. That means we break the electronic connectivity between the formaldehyde-hydrogenase and the hydrogenase, and we expect the enzyme to be dead, and it is. And if we mutate HIGB3, now it becomes interesting, because now we disrupt the connectivity between the repeating subunits. And that means, in theory, we still have a minimal active complex, but this is now reduced by activity drastically. So what does this mean? So if we now elongate this molecule in our mind, what do we get here? We get something incredible. We get an enzymatic decorated nanowire. So we have a long iron-sulfur chain transferring a long protein. We have tons of hydrogenases sitting here. We have formaldehyde-hydrogenases here. And I like to think of it similar to photosystems, which have light harvesting complexes, as a hydrogen harvesting antenna. Hydrogen is very rare where these bugs live. So this is a hypothesis. We don't know if this is true, but it's a captivating idea. So we need to prove this now. How did we do this? We took our protein, run it over analytical gel filtration, and roughly divided it into 4 fractions, and determined the activity of every of these fractions. And we could see, indeed, that the longer fractions, basically already the void, more or less, is the most active. And when we go to smaller fractions, the activity decreases as well. In the interest of time, I can't show you, but we have the data that the lower fractions are basically 3 repeating subunits, 6 repeating, 9, and then larger filaments. And this assembly in steps of 3 seems to be important to enhance the activity of the complex even further. So we also tested this now in vivo. So if we look closely, there's one iron-sulfur cluster which is mainly there to make the connection between the repeating subunits. Here in HIGB4, this iron-sulfur cluster. If we mutate it, there's still connectivity between the hydrogenase and the formatter hydrogenase here, but not like within the filament. And this protein still forms a larger complex. That means it's structurally intact. It can still oligomerize. And we also tested it in vivo. It has the same compensation phenotype, more or less, as the wild-type protein. And the activity, however, is drastically reduced. So this means if we maximize the filament length, we also maximize the activity of our enzyme. So this is not anymore a hypothesis. Long polyferrodoxin electron wires exist. Now comes the questions which we asked ourselves, but not really dared to answer it. But you can be sure our referees did. The question was, how is the situation in vivo? How is it in the cell? Do we have a long filament or do we have multiple short filaments that could still be active? And the only way how we actually can answer this question is because fluorescence microscopy in an anaerobe doesn't work very well. GFP needs oxygen. So we need to see inside of the cell, and this we can only do with cry-electron tomography. And I cooperated with Ben Engel, who was at that time at the Helmholtz Center in Munich, and is now in the Biocentrum in Basel, and we carried out cryo-FIPSAM to basically cut out small slices of acetogen cell frozen on grids, and these slices are electron transparent to the electron beam, as you can see here. And we saw some filament here at the top of the cell. And this becomes more clear in the top tomogram. You can see here in black dots, these are the ribosomes. Here you see the S layer, the bacterial coat, and now it becomes more clear. Here in gold, at the tip of the cell, we saw filaments. We saw filament bundles, and they are huge. And something like that, I've never seen in any tomogram of any other bacteria so far. And to verify that this is indeed HDCR, we also carried out a tomography of HDCR knockout cells. And these filaments, they were completely absent. They have a diameter of roughly over 200 nanometer. And to just get a dimension idea, you can see here a ribosome. It fits comfortably inside it. So these large ring-shaped superstructures are our HDCR filaments. So what did we learn about HDCR? We discovered an electron-decorated nanowire. Protein connectivity and electron coupling along the filament are important to maximize its activity. And in vivo, HDCR does not only form filaments, but this large ring-shaped superstructure. They are membrane-associated, and they contain thousands and thousands of iron-sulfur clusters. This is actually a huge effort for the bacterial cell to build something up like that. So the question now is, why they are anchored to the membrane? What is the physiological function of this? In short, we don't know yet, but we at least allowed a little to speculate. And I think this cellular superstructure might have something to do a little bit with organization of the metabolism. So one could maybe make an analogy to a pyrenoid in an algae cell or maybe a carboxysome in a cyanobacterium. But instead of a carbon concentration mechanism, I proposed a former centerpiece of a formate concentration mechanism. Because CO2 diffuses into the cell and is trapped as formate. And for the Woodley-Jungdahl pathway to work efficiently downstream, you need quite high intracellular concentration of formate. I think this superstructure is one of the adaptation, how this is actually managed. And the next obvious question is this the odd one out, or are there more nanowires to be discovered? And here I can give you at least part of an answer. There are more nanowires. Of course there are. Here you can see a structure of a tungsten-containing aldehyde oxidoreductase. It's an unrelated enzyme, but you can basically see here, it forms the filament, and if we superpose the core structure, the PIC-D4 with the RAA, they're basically spot on identical. They also have this double ferrodoxin fold in the C-terminal helix. And if we delete the C-terminal helix, we can see we dissociate the filament in the minimal repeating units. So the same as in HDCR. But there's one difference. The difference is here. The electron input module is not associated along the filament. It has a cap. And this cap makes it a different nanowire. So we have the HDCR filaments. They are very long. And we have the capped nanowire. They are rather shorter. One of the reasons probably is that capping is introduced kind of nucleation mechanism. And if you have multiple nucleation mechanisms, you tend to not form very long filament. And the second thing is, HDCR has two types of Higg-B proteins, and the AOR only one. So at one point, there might be some steric conflict between the enzymes if it filaments around itself. So now with modern tools in structural biology, fold-seq, phylogenetic analysis, we're in a big hunt on more nanowires. And I can't present you the data yet, but we have found a lot of them. Some of them are here. So again, we have the connection of the Wood-Leon-Gar pathway from bacteria to also archaeal lifestyle. So I also brought you a second short story. And this is on the central respiratory enzyme of acetogens, the electron bifurcating hydrogenase, that uses the oxidation of hydrogen to produce low potential ferrodoxin for respiration. But stop. No, it can't. It is actually a thermodynamic forbidden reaction. It's an anorganic reaction. You cannot do this. The potential doesn't allow it. You cannot go from minus 400 millivolts to minus 500 millivolts. But the cell still does it. And how does it do it? The trick is electron bifurcation. It's nature's way to catalyze anorganic, so thermodynamic forbidden reactions. And this is by carrying out a piggyback rate. So at the same time, you catalyze an exergonic reaction, something where energy is freed. And this drives the reaction, which is not allowed. So by direct coupling of anorganic and exergonic reactions, you can create reactions which are thermodynamically not feasible. So how does this enzyme do it? What you need to know, there are actually 4 phylogenetic unrelated classes of bifurcation enzymes. 3 of them, they work by a classical way of bifurcation. And they use a special property of flavin cofactors. Flavin cofactors can carry out 2 to 1 electron conversions. And you can just say, okay, bifurcation enzymes perform electron magic. They take a 2 electron pair taken on a flavin and split it into 1 electron ways, and one goes high and one low potential. That's it. That's the basic principle what they do. And to be able to do this, they need to have a special stabilized anionic semiquinone. Just important, this exists and it's stabilized by special residues. And the second thing, which the classical bifurcation enzyme usually have, is they have a second FAD. And that is usually there to just bind a pyridine cofactor. And if we now look at the cofactor composition, what we would get from our electron bifurcating hydrogenase, we realize this doesn't add up. There are a lot of iron-sulfur clusters, 4 iron-sulfur clusters, 2 iron-sulfur clusters, and we have 1 FMN cofactor. And just based on sequence analysis, I can tell you, this has no hallmark features of a stabilizing anisemiquinone. So where's the high and where's the low potential branch? How could bifurcation possibly work in this enzyme? And being a structural biologist, we determined the structure of the enzyme. And we also determined it on fully reduced conditions, we determined on oxidized conditions and neutral conditions. And what you can see here in blue, again, hydrogenases, and here in orange and red, basically an NAD dehydrogenase-like domain, basically here a thyrodoxin-like domain, there's another thyrodoxin-like domain, and here is something which reminds you of the core of Higg-B. This is true. This is a linked ferrodoxin-like domain. And what we can again understand here, this domain is probably here to bind ferrodoxins, because this is what ferrodoxins-like domains do. So if we now look at the electron pathway, we see here the site of hydrogen oxidation. It's carried out by a special cofactor called H cluster. The electrons go into it, they go to the FMN, then they can go distance-wise barely to B2 and C1, and that's it. Here, where the ferredoxin reduction side would happen, there's a huge gap, over 13 angstrom. No way that in the time of the universe, an electron can bridge this distance. And here at the FMN, surprisingly or not surprisingly, I don't know, we see an NAD bond. I say surprisingly because we didn't add it, and this means it's co-purified. So this comes a little bit of a surprise because usually FMN-bound enzymes don't co-purify with cofactors. Sometimes they even lose the FMN. And we went to the lab and tested this. So if we reduce our enzyme, we can basically have a high affinity for the FMN. And if we oxidize it, this affinity is gone. So now we look at the vicinity. So what could potentially trigger this? Of course, here it is two clusters. And if we mutate either one of them, this effect is gone. So what can we conclude? We can conclude that this is a redox-dependent switch. So the special environment of the FMN cofactor makes it to modulate that it can either bind in a reduced form the pyridine nucleotide or not. And if we now look again close in the vicinity of the FMN cofactor, we don't see anything that could bind anion SYNMIKRO, but instead we see here this aspartate. This aspartate can stabilize a different kind of quinone, neutral SYNMIKRO. But this is not that important. What is important is that by comparing the different structures, NAD plus bound, NADH bound, and apostate, we can see this movement. And by moving, it kicks out this second domain, the SYT domain, and this 2 iron 2 sulfide cluster bridges the distance from 13 to roughly 15 angstrom. So you could say it carries an electron from one side to the other. And this other side is here the SIG-B3 big 4 cluster, and this is where we can complete the reaction now. You can see in ND simulation that here is the ferredoxin binding site, and the enzyme can now rapidly transfer the electrons to the low potential ferredoxin, at least if it can catch it. So what can we conclude? We can conclude that this enzyme class is certainly very different from classical bifurcation enzymes. It works by affinity modulation and by kinetic gating to distinguish between the high and the low potential pathway. So it's a unique class of bifurcation enzymes. And if you know your textbook literature, this reminds me a little bit of complex 3 or cytochrome B6F and mitochondrial chloroplast, where the risky iron sulfur proteins also does a similar movement in the Q cycle of the enzyme. And this is of historical importance because this is the reaction where the bifurcation was for the first time discovered. So this kind of closes the circle. So I want to end with a side note. And this is this bifurcation module, the so-called HYTBC module, can be replaced with other enzymes. It can exist with the electron bifurcating hydrogenase, but it can also bind to different enzymes. Here you see another of these enzymes. It's called the SDN transhydrogenase. So it converts NADH to NADPH and ferroxide. And the details are not important, but I want you to grab the concept of modularity. So we can play molecular Lego. We can take one type of enzymatic activity and combine it with a bifurcation module and get a bifurcating enzyme. And we kind of take it to the extreme in our lab, and we did so there was some phylogenetic analysis done by the Adams group, and we took core members of different phylogenetic branches and looked at them. And we basically see the SYTBC modules are involved in all kinds of anaerobic reactions, from redox balance to CO2 fixation to bioremediation. So this is a bifurcating HTCR type complex. They also do exist. And this is absolutely amazing. So anaerobic microbiology is very different to what you know from aerobic respiration. And the key concepts can be only understood by carrying out redox-controlled cryo. You can study very nicely cofactor binding. So you see a very oxygen-sensitive tungsten cofactor. And you can even see small details. You can see the change of iron-sulfur cluster coordination dependent on the redox state of the enzyme. So there's a lot of biology to be uncovered if you look closely. And you need a whole combination of integrative structural biology, spectroscopy, biochemistry, and microbiology to go at the bottom of this very fascinating enzymes. So I hope in the last few minutes I could indulge you into the principles of anaerobic metabolism. It's different, but it's very fascinating. We have modularity. We have redox-controlled conformational changes. We have a kinetic gating mechanism. And we have overall a lot of redox cofactors. And by studying this, we understand how this functions. We can kind of come to an understanding of ancient metabolic pathways, but also we lead the way in biotechnological applications in synthetic biology. And I hope I could convince you that this is very well worthwhile to do, that the study of biological CO2 fixation is important. And in the end, I now want to thank my team. A lot of the work has been carried out by my PhD student Anush Kumar, also by Fidel, and my cooperation partner, Stefan Bohn at the Helmholtz Center Munich, Wille Keiler for in-deep simulation, Volker Müller, who introduced us into genetics, Ben Engel, who introduced us into cryo-ET, Francis, with whom we're doing cross-linking MS. So you see, a lot of methods come together in the integrative spirit. And I'm happy to take your questions now.